All right, thank you for watching my presentation. It is on comb jellies and gastroid fungi, otherwise known as stinkhorns. And if I zoom into my little chart here, you'll see that comb jellies do come from the animal family and stinkhorns do come from the fungi family. My first slide is about my theme that I chose, and that is cell division and development. Now, what makes these two organisms so interesting is how differently they grow compared to pretty much everything else that scientists have studied. So most organisms follow a pretty strict rules about cell division. Cells split evenly, they develop symmetrically, they grow, at a steady pace for the most part. But these two organisms are very quite different than what we are used to. Comb jellies, they divide their cells in ways that are very, very weird. And stinkhorn fungi, they kind of just pop out. And then as soon as they come out of that mycelium, they will grow from, what you would call a baby, so to speak, to a full-grown mushroom in just a few hours. So these are some really, really interesting creatures, and I would love to learn more about it with you in this presentation. All right, so what makes a comb jelly special? Number one, they are marine animals that swim using 8 rows of hair-like cilia. Remember, we talked about cilia in first semester of Gen Bio. Those are the hairs that kind of go with like a waving rhythmic pattern forward and backwards, and this actually allows them to move. They're also transparent, which is kind of cool because you can see, all the insides, especially the nervous system. They catch their prey using these sticky cells. They're found in oceans, and I actually found an article that talks about that there is some belief that comb jellies are the furthest ancestor of all creatures in the world. So I did put that in the reference section if anyone wants to check that out. The other cool thing is they can age in reverse. That's actually a really cool fact. They can go from adult to back to, for lack of a better word, a childlike state, and then to adult again. And as I said before, they have really unique cell division patterns because their embryos divide very differently than most other animals. They do build their bodies without using common animal development genes. For example, Hox genes, they do not have Hox genes. And to be honest, they use some genetic tools that we don't actually understand yet. The science is not quite caught up to everything that these creatures can do. For the stinkhorn, what makes the stinkhorn stink? So, again, these are tall, distinctive mushrooms or fruiting bodies, and they emerge rapidly from egg-like structures underground. And the reason why they're called stinkhorns is because they literally smell like rotting flesh. And that rotting flesh attracts insects, and those insects spread the spores, and that's how they reproduce. They're often found in rotting woods like fallen trees, and they can grow to full size in just a few hours. As I said before, the way they divide their cells is very rapid, and when it's time, they can go from laying flat on the ground or under the soil to completely all mature fungi. They are different from other fungi because most other fungi grow slowly. It takes sometimes weeks to go from just the mycelium to a fully fruited mushroom, but not these guys. Comparison time. So cell division. Comb jellies have holoblastic cleavage, which means it is full, complete cleavage, whereas stinkhorns have localized cell expansion through Turgor pressure. Comb jellies, as we said before, they're kind of weird with the pattern of how their cell division happens. They have radial and spiral cleavage. And the stinkhorns, the cells only proliferate during fruiting body maturation, which is, again, that very short few-hour time period. Comb jellies do use morphogens, especially in embryonic development. Stinkhorns, on the other hand, do not at all. They actually use other triggers like moisture and temperature that tell them when to begin their cell expansion. As I mentioned in the last slide, comb jellies don't have Hox genes. They are not like us in that case. Instead, they rely on different genetic pathways that kind of go beyond the scope of this particular presentation, maybe in our second or third vlog. I'm not sure yet. All right, comparisons continued, some similarities and differences. Both the stinkhorn and the jelly do use rapid cell changes for development. They have each evolved in their own way to survive in their environment, and they both differ from typical development that we see in animals and other fungi, which is kind of cool. The differences, comb jellies use, as I said, unusual patterns of cell division in early development, and they build their bodies without typical animal development genes. Whereas stinkhorn, they grow by rapid cell expansion over just a very short amount of time rather than division, and they can emerge fully formed in just hours. And for research impact, I talked about how comb jellies challenge what we know about animal development. They are very unlike the majority of other animals. They use a unique set of developmental genes and proteins to control how their body plan is made. And then they use like their own regulatory networks. And part of this is because they split from other animals so early in the evolutionary process. As I said, there's some evidence that they were the first or the farthest back creature that we found. And stinkhorns, on the other hand, they rely on environmental triggers like temperature and humidity. So they have kind of alternate growth strategies, even among fungi, not just among creatures in general, but they rapidly develop very, very quickly. So that is different than a lot of other fungi. If you've grown something at home, like portobellas, for example, just some other mushroom, you'll know that once that mycelium starts to spread, you may still have another week or two before you get fully formed mushrooms. And that's not the case with stinkhorn fungi. Future questions. Could we use these creatures for biomedical applications? This is actually a question I found online. I thought was a really good one. Like, what do we see in stinkhorn fungi with their rapid cell development, and what can we learn from the jellies that may help us in the future, whether it is biomedical applications for humans or for other creatures and animals? Here are my references. I used a mixture of DOI.org sources, which I always love going to, and also some secondary sources. And the image credits are also in here as well. Lastly, I use Microsoft Copilot. The prompts I used were comb jelly cell division, stinkhorn cell division, similarities and differences of comb jelly and stinkhorns, and show cell division comparison chart.